investigating the role of self-antigens in activating B cells in atherosclerosis using transgenic B cell receptor mouse models. Atherosclerosis is an inflammatory disease of the large and medium-sized arteries that is characterized by deposition of oxidized low-density lipoproteins, or ox-LDL, within the vessel. While specific self-antigens are not well characterized, antibodies against LDL, ox-LDL, and ApoB show that the immune response, specifically B cells, is involved in atherogenesis. The role of B cells in atherosclerosis is B cell subset specific, with follicular B cells being pro-atherogenic and B1 and marginal zone B cells serving as protective, at least partially due to their ability to produce natural IgM. It is currently unknown whether the B cell receptor can recognize ox-LDL and whether or not atherosclerosis can alter the state of anergic B cells. We took advantage of 2 transgenic mouse models in order to investigate the role of the B cell receptor repertoire. The ARS-A1 mouse model has a transgenic B cell receptor that results in only the production of anergic B cells. And this is due to constant B cell receptor stimulation, and the other MD4 mouse model consists of a transgenic B cell receptor that can only recognize the HEL ligand, and thus produces unresponsive B cell receptors. However, these are not considered anergic. In order to induce atherosclerosis, MD4 and ARS-A1 transgenic mice along with wild type were injected with PCSK9 AAV and fed a high-fat diet for 28 weeks. Chow-fed mice were also utilized as homeostatic controls. In order to assess production of ox-LDL-specific IgM, a homemade ELISA was generated to analyze the plasma concentrations. In chow conditions, we found that there were no significant differences between wild-type MD4 and ARS. However, in high-fat diet atherosclerotic conditions, MD4 produced significantly less ox-LDL-specific IgM compared to both the wild-type and the ARS. When looking at the activation levels of B-cell subsets, we found that the activation of B cells is specific to different B cell subsets. Interestingly, ARS have increased activation in their B1, B1A, and B1 and marginal zone B cell subsets. In summary, you found that wild-type ARS and MD4 produce similar amounts of ox-LDL-specific IgM in homeostatic conditions, indicating that the production of ox-LDL-specific IgM can be produced constitutively, at least partially independent of the B-cell receptor repertoire. However, in atherosclerotic conditions, the transgenic B-cell receptor leads to differences in production of ox-LDL-specific IgM, indicated by the significant decrease of MD4 production of ox-LDL-specific IgM. This potentially indicates that the inflammatory environment of atherosclerosis can influence activation of B cells, and that this may be at least partially dependent on the B cell receptor repertoire. When looking at B cell subset activation, we found that different B cell subsets express different levels of activation in atherosclerotic environments. Interestingly, IRS marginal zone B cells, total B1 B cells, and B1A B cells have significantly higher levels of activation compared to wild type. Further investigation will focus on how the B cell receptor repertoire can influence activation of specific B cell subsets and maturation into antibody-producing plasma cells and its role in atherosclerosis.